Hi, I am Bulbul Ahmed, PhD student of Integrative Bioscience, Tuskegee University. I am preparing this video for a partial fulfillment of the final examination of the molecular biology course. My subject of this video is central dogma from DNA to protein. First, we have to know what is central dogma. The original postulates that genetic information can be transferred only from nucleic acid to nucleic acid, from nucleic acid to protein, or from DNA to DNA to DNA to RNA, or from RNA to protein. Where these types of discussion is happened, this is central dogma. First, we have to know what is the structure of DNA. Deoxyribonucleic acid is a nucleic acid that contains the genetic instruction of the development, functioning, growth, reproduction of all the organisms. All organism is belongs DNA, and DNA is the main part which continue the hereditary elements from one species to another, one organism to continuous to this next offspring's organization. DNA serves as the store of hereditary information within a cell, and the carrier is this information from generation to generation, hence named the major biological block of life. DNA and ribonucleic acid or nucleic acid are one of the four major types of macronym molecules that are essential for all known forms of life, namely protein, lipid, complex, and carbohydrate. Most DNA is in the cell nucleus, where it was called nuclear DNA, but a small amount of DNA can also be found in the mitochondrial DNA. Okay, the difference between DNA and RNA is the RNA contains the sugar ribose, where DNA lacks of oxygen atoms, and RNA has the nucleobase uracil, while DNA contains thymine. Building blocks of DNA. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The four types of nitrogen base are adenine, guanine, thymine, and cytosine. Adenine is termed as A, thymine T, guanine G, and cytosine C. The order of these bases in what determines DNA's instruction or genetic code. Nucleotides are the building block of nucleic acids. They are composed of three subunit molecules. Four nitrogenous bases in DNA are adenine, cytosine, guanine, and thymine. 5 carbon sugar, ribose and deoxyribose, and phosphate groups are shown in the DNA structure. Now, I want to discuss on nucleotides and bases. The sugar and phosphate group make up the backbone of the DNA double helix. DNA double helix is first introduced by Watson and Crick, while the bases in the middle, a chemical bond between the phosphate group of the nucleotide and the sugar of a neighboring nucleotide holds at the backbone together. Chemical bonds, hydrogen bonds between the bases that are across from one another hold two strands of double helix. Bases are the part of DNA that stores information and gives DNA the ability to encode a person's visible trait. Adenine, guanine are purine base. These are structure composed of a 5-sided and 6-ring. Cytosine and thymine are pyrimidine, which are structures composed of a single 6-sided ring. During the formation of DNA nucleotides are bonding to each other by forming a bond of linkage between OH group of the sugar of one nucleoside and the other hydroxyl group, phosphate group of adjacent nucleotide. During the bonding, the OH groups are participating into water, hence termed a condensation or dehydration process. The bonding process involves joining of hydroxyl group of 3' carbon of the sugar group and 5 carbon of the adjacent phosphate group. The links between the nucleotides are called phosphodiester bond. Phosphodiester bond is a covalent bond in which two hydroxyl groups form ester linkage to the same phosphate group. Joins adjacent nucleotides in DNA to form the backbone of the DNA strands. Another forces that connect nucleotides of each other is the force that occurs across strands, termed as base pairing. Base pairing bonds occurs when one purine bonded to another pyrimidine. During base pairing, adenine always binds to thymine in DNA and uracil in RNA, while guanine binds to cytosine. This type of bonding between two members ring and one member ring is essential for the maintenance of the uniform thickness and distance. Now I want to discuss on DNA double helix formation. DNA double helix arises from the chemical and structural features of its two polynucleotide chain. Simply, it is formed when one strand and its complement strand are base pairing each other. That means two chains are held together by hydrogen bond between the base GC and AT on the different strands. All the bases are on the inside of the double helix and the sugar phosphate backbones are on the outside. This complementary base pairing enables the base pairs to be packed in the energetically most favorably arrangement in the interior of double helix. In this arrangement, each base pair is of similar weights, thus holding the sugar phosphate backbones are equal distance apart along the DNA molecules. To maximize the efficiency of base pair packing, the two sugar phosphate backbones wind around each other to form a double helix. With one complete turn, 10 base pairs. In natural DNA, and almost always hydrogen bonds with T and G with C forming AT and GC base pair, often called Watson-Crick base pairs. The impact of the double helix. Double helix bond is the bond. We know that this is a bond which bind the by TAT-CG base pair. The structure of DNA unlocked the door is understanding many aspects of DNA's function, such as how it was copied and how the formation is carried. Was used by the cell to make protein. This tells that if you have a double helix, the strands will separate will be a template for double helix during hereditary. The original postulate that genetic information can be transferred only from the nucleic acid to nucleic acid and from nucleic acid to protein, that is from DNA to DNA, from DNA to RNA, and from RNA to protein. Central dogma may be describes on three process, including DNA replication, DNA transcription, and DNA translocation. First, I want to discuss on DNA replication. DNA replication within the nucleus must be replicated in order to ensure that its new receipts are the correct number of chromosomes. The process involved multiple proteins called replication enzymes and RNA. In eukaryotic cells, such as animal cells and plant cells, DNA replication occurs in the S phase of interphase during the cell cycle. The process of DNA replication is vital for cell growth, repair, and reproduction of the organization. Replication, the first step of the replication is fork formation. The double-strand molecules must be unzipped into two single strands by breaking hydrogen bond between base pairs facilitated by known as DNA helices. DNA helices disrupts the hydrogen bonding between base pairs to separate the strands into Y shape known as the replication forks. The separate strands stand as the template of replication to begin. DNA is directional in both strands, signified by 5' and 3' end. The notation signifies which side group is attached in DNA backbone. The 5' end has a phosphate group attached, while the 3' ends has a hydroxyl group attached. This directionally is important for replication as it only progresses in the 5' to 3' direction. However, the replication forks is bidirectional. One standard is oriented in the 3' to 5' direction, while the other oriented 5' to 3' strands. The two sides are therefore replicated with two different processes to accommodate the directional differences. Second step of DNA replication is binding, primer binding. A short piece of RNA called a primer binds to the 3' end of the strands. The primer always binds at the starting point of replication. Primers are generated by the enzyme DNA primase. Next step is elongation. Enzyme known as DNA polymerase are responsible creating the new strand by a process called elongation. Polymerase 3 is the main replication enzyme, while polymerase 1, 2, 4, and 5 are responsible for error checking and repair. DNA polymerase 3 binds to the strands at the site of the primer and begins adding new base pair complementary to the strand during replication. In eukaryotic cells, polymerase alpha, delta, and epsilon are the primary polymerase involved in DNA replication. Because replication proceeds in the 5' to 3' direction on the leading strands, the newly formed strand is continuous. The next step is termination. When both of the continuous and discontinuous strands are formed, the enzyme called exonuclease removes all RNA primes from the original strands. These primers are then replaced with appropriate bases. Another exonuclease proofreads of newly formed DNA to check, remove, and replace any error. Another enzyme called DNA ligase joins Okazaki fragments together, forming a single unified strand. The ends of the linear DNA present a problem as DNA polymerase can only be nucleotides in the 5' to 3' direction. The key enzymes for the replication is we can say first DNA helices unwinds and separates double-stranded DNA to form the replication fork and breaking hydrogen bonds between nucleotide pairs in DNA. Next, DNA primase. It is a type of RNA polymerase that generates RNA primer. Primers are short RNA molecules that act as templates for the starting point of DNA replication. Next, DNA polymerase. Synthesis of new DNA molecules by adding nucleotides to leading and lagging DNA strands. Topoisomerase or DNA gyrase unwinds and rewinds DNA strands to prevent the DNA from becoming tangled or supercoiled. Next, exonuclease this group of enzyme that remove nucleotide base from the end of a DNA chain, and then the DNA ligase joins DNA fragmentation together by forming phosphodiester bonds between nucleotides. Next, I want to discuss on transcription. Transcription is the first step of gene expression. In which information from a gene is used to construct a functional product such as protein. The goal of transcription is the main RNA copy a gene DNA sequence. For a protein coding gene, the RNA copy of transcript carries the information needed to build a polypeptide protein or protein subunit. Eukaryotic transcripts need to go through some processing steps before translation into protein. In transcription, a region of DNA opens. One strands, the template strands, serves as a template for synthesis of complementary RNA transcription. The other strands, the coding strands, is identical to RNA transcription in sequence, except that it has uracil base in place of thymine base. And now I want to discuss on stages of transcription. Here the steps are initiation, elongation, termination. Initiation. RNA polymerase binds to sequence the DNA called the polypromoter found near the beginning of a gene. Once bound, RNA polymerase separates the DNA strands, providing the single-stranded templates needed for transcription. The promoter region comes before and slightly overlaps with the transcript region. Those transcription is specifies. The DNA opens in the promoter region so that RNA polymerase can begin transcription. And next is elongation. One strands of DNA, the template strands, acts as a template for RNA polymerase. As it reads this template one base at a time, the polymerase builds an RNA molecule out of complementary nucleotide, making a chain that grows from 5' to 3'. The RNA transcript carries the same information as the non-template coding strand of DNA, but it contains the base uracil instead of thymine. The synthesized RNA only remains bound to template strand for a short while. Then exists the polymerase as the dangling string, allowing the DNA to close back up and form a double helix. And then I want to discuss on termination. The sequence is called termination signal that RNA transcript is complete. Once they are transcript, they cause transcript to be released from polymerase. And then I want to discuss on translation. Proteins are key molecule building block for every organism on Earth. Basically, a gene is used to build a protein in a two-step process. Transcription stage in which the DNA sequence of a gene is rewritten in the form of RNA. The translation in which the mRNA is decoded to build protein that contains a specific series of amino acid. The genetic code. During translation, a cell reads the information in a messenger. This may be mRNA, and uses it to build a protein. To be a little more technical, an mRNA does not always provide instructions for a whole protein. Instead, we can confidently say is that it always encodes a polypeptide or chain of amino acid. In genetic code table is three-letter sequence of mRNA nucleotides corresponds to a specific amino acid or to a stop codon. UGA, UAA, and UAG are stop codon. AUG is the codon for methionine and is also the start codon. In an mRNA, the instructions for building a polypeptide are mRNA nucleotides. There are 61 codons of amino acid, and each of them is read to specify a certain amino acid out of 20 commonly found in proteins. One codon, AUG, specifies the amino acid methionine and acts as a start codon to signal the start of protein construction. And then I want to discuss on transfer RNAs. Transfer RNAs or tRNAs are molecules bridges that connect mRNA's codon to the amino acids they encode. One end of each tRNA has a sequence of 3 nucleotides called an anticodon, which can bind to a specific mRNA codon. The other end of the tRNA carries the amino acid specified by codons. Then I want to discuss on ribosome. Ribosomes are the structures where polypeptides protein are built. They are made up of protein and RNA, ribosomal RNA or rRNA. Each ribosome has 2 subunits, a large one and a small one, which come together around the mRNA, kind of like the 2 halves. The ribosome provides a set of handy slots where tRNAs can find their matching codons on the mRNA templates and deliver their amino acid. These slots are called the AP, AP, and E sites. Not only that, but also ribosome also acts as an enzyme, catalyzing the chemical reactions that link amino acids together to make a chain. Now I am discussing on steps of translation. First, initiation. Initiation. The ribosome assembles around the mRNA to be read, and the first tRNA carrying the amino acid methionine, which matches the start codon AUG. This setup, called the initiation complex, is needed for translation to get started. Second step of translation is elongation. Elongation is the stage where the amino acid chains get longer, and elongation, the mRNA is read 1 codon at a time, and the amino acid matching each codon is added to a growing protein chain. Each time a new codon is exposed, a matching tRNA binds to a codon. This existing amino acid chain, that means polypeptide chain, is linked onto the amino acid of the tRNA, which via a chemical reaction, this mRNA is shifted 1 codon over the ribosome, exposing a new codon for reading. And then termination. Termination is the stage in which the finished polypeptide chain is released. It begins when a stop codon, UAG, UAA, or UGA, enters the ribosome, triggering a series of events that separate the chain from its tRNA and allow it to drift out of the ribosome. After termination, the polypeptide may still need to fold into the right 3D shape, undergo processing, get shipped to the right place in the cell, and combine with other polypeptides before it can do its job as a functional protein. And now I want to discuss on gene cloning, DNA cloning. DNA cloning is the process of making multiple identical copies of a piece of DNA. In DNA cloning procedure, the gene or other DNA fragments of interest is first inserted into a circular piece of DNA called plasmid. The intersection is done using enzymes that cut and paste DNA, and it produces a molecule recombinant DNA and DNA assembled out of fragments from multiple sources. The recombinant plasmid is then introduced into bacteria. Bacteria carrying the plasmid are selected and grown up. As they reproduce, they replicate the plasmid and pass it on their offsprings, making copies of DNA contain. Steps of DNA cloning are cutting and pasting DNA, bacterial transformation and selection, protein production, and there is also DNA sequencing, Taq polymerase, PCR primers also. So this is important for DNA for central dogma. Thank you very much for watching the video.